Thank you. All right, so last summer, I was working on a project at school involved screening Arabidopsis mutant lines for transgene, and it was a lot of waiting around and a lot of repetitive PCR, and I was kind of not stimulated enough. So for better or worse, this summer, I decided I wanted to experience a lot of different things. That said, everything I did involved developing different methods for manipulating meiotic recombination in Mays. Right, so what is meiosis? So if you remember from high school bio, meiosis is a cell division process that occurs in sexually reproducing organisms that halves the chromosome number so that between generations the chromosomes are conserved. And the most important part of meiosis is when the chromosomes line up in the middle of the cell right before they're going to be split into two different daughter cells. And during this process, the chromosomes can often get pretty tangled. So cells usually cut the chromosomes to stop them from being tangled, and then another enzyme will repair that cut by either attaching the DNA back to the same chromosome it was attached to in a non-crossover event, or to the other chromosome in a crossover event. And as you'll see, the crossovers are the most important part of recombination from my perspective. So why are crossovers important? So consider an example of two maize plants. You have a really tall maize plant, and you have a maize plant with really sweet corn. You cross them together, and the daughter maize has both of those genes. But when you try and produce offspring from that daughter maize, the chromosomes are just going to segregate again, and you're only going to get tall plants and sweet plants. But if you have crossover between the two genes that control those loci, you can get daughter maize from that daughter maize that has both traits. So our goal is to accelerate that process because maize takes 6 months to grow. So if we can reduce the number of generations that it takes to get two traits we want together, that would be great because we don't have to wait 6 months. And then also it would be great if we could target specific genes for crossovers, like tall maize and sweet maize. And if we can increase the overall number of crossovers, the genetic diversity of the daughter population will also be much larger. So how do we study meiosis? Well, the best way is by looking at proteins. So these are the two proteins that I ended up looking at, MLH3 and Hi10. So they both are involved in promoting crossovers, and they co-localize on the DNA where crossovers occur. And they're highly conserved between species. So Hi10 was actually first studied in humans, and MLH3 was first studied in bacteria. So we sort of know what they do from that. So here you can see a picture of Hi10 localizing on Arabidopsis chromosomes during meiosis. But we can't just make this image just by identifying the proteins. We actually have to tag them with something. So the way that we do that, there's two different ways. The first is by making antibodies for that protein. So in your body, antibodies are used by your immune system to tag in foreign objects to be destroyed by your immune system. But we can manipulate this to be used in microscopy by injecting the protein that we want to be tagged into an animal, and that animal will produce antibodies to that protein. We can harvest the antibodies, and then we can use them in microscopy. The other method is by attaching the GFP sequence to a certain protein that we want to image, and then when that protein is expressed, it will glow green, as you can see here. So antibodies are great in the fact that they can be made really fast. Like once you have a recombinant protein, all you need to do is inject it into the animal, and then a couple weeks later, months later, you'll have antibody, and you can use that forever. And you can use it on pretty much anything you want within the same species, even between species. But the problem is it's really unreliable. Like you don't know if an antibody will work until you use it. GFP is much more reliable. Like we know it works, and you can actually use it to image things in vivo. But the problem is you have to transform the plants you want one by one. So the first thing I did was make an antibody for MLH3. So I started out by trying to find the MLH3 gene in maize. So unlike Arabidopsis, the maize database is not super well annotated, so I couldn't just Google MLH3 maize and find something. So I had to blast the Arabidopsis sequence into the maize database and take the best hit. And then after that, I blasted the best hit back into the Mays database to find the unique regions, because to make an antibody, we want the antibody to bind to the regions that are only in MLH3, not to things that are conserved between multiple genes. Once I found those 2 unique regions, there ended up being 2 in MLH3. We called one the N term because it was near the front of the protein. We called the other the C term because it was near the end of the protein. I made primers for those sequences and amplified them, transformed them into this expression vector. This expression vector has the T7 promoter, so the gene that is in between the promoter and the terminator will be expressed when you induce it with lactase, or IPGG, which is a similar structure. And that will get the bacteria to produce the protein when they're at the right growth stage. And then we can harvest the protein, purify it, and send it to the company to inject into the rabbit or another animal that will make the antibody for us. So there were 2 important parts in verifying that I did this correctly. The first was making sure the sequence that I inserted was correct. And the quick way to do this was do a PCR. And you can see it matches the control. And then if that works, you usually send it off to sequencing, which we've done. And then the second is to make sure that the protein is actually expressed. And we do this by an SDS page on the purified protein. And you can see that all 3 of the proteins match about their expected size. All right. So the second thing I did was make a GFP mutant. So this started out in a very similar manner. I had to blast the known high 10 Arabidopsis gene into the Mays genome and look for the best hit. And unlike MLH3, there were actually multiple pretty OK hits. So I was curious about them. There was obviously one best hit, but there were 2 that were not completely dissimilar. So I decided, since I had some extra time, I would figure out where those genes came from. So the first thing I did was a multiple sequence alignment between all 3 of the high 10 homologs that I found, or paralogs that I found. So you can see in the red is the region that's highly conserved between all of them. The second one has a deletion relative to the one I thought was canonical, and the third one has a deletion at the beginning, as well as a large extra region at the end. So I actually blasted this large extra region against the Mays database, and I found that it was highly homologous to an arginine methyltransferase. So I wanted to know where these genes came from. So I made a phylogenetic tree with those genes, as well as high 10 from a bunch of other plants, as well as methyltransferase from a bunch of other plants. And so what I found was, since the Mays genes all cluster, that they probably all originated in the most recent whole genome duplication event in Mays, which was about 11 million years ago. But the fact that one of the high 10s was also quite close to the methyltransferase makes me think that this protein is a chimera between the two proteins. And that these proteins are both expressed according to the database. So it's possible that they could have some involvement in crossovers, but we don't have enough information right now. So back to the main project. So after I identified the best homolog, I made primers for its cDNA as well as primers for its promoter, because it has to, in order to be a legitimate GFP mutant, it has to express at the same level as in wild type. So I transformed all of those into the vector, which is p-enter vector, using restriction digests. And then after everything was in, we were going to flip it into the transformation vector, which will be used to transform the Mays, which takes about 3 months. So here you can see what the vector should look like if everything's inserted. You have your restriction sites where it will insert the genes of interest, and the arms that will be used to flip the vector into the transformation vector. And so to verify that everything was where I thought it was, I did 2 different PCR reactions. The first was the promoter primer going forward and the gene primer going reverse. And this, we expected a band around 2.9 kb. And you can see both colony 7 and 8 have it, as well as the control, which was from a previous cloning. And then the second was to make sure that GFP was in and also in the right way, because of these 2 restriction sites, it could have also been inserted backwards. So we expected a band around 3.6 with the promoter forward and the GFP reverse, and that's what we had. So looks like I have this vector in the bacteria that I want. All right, so why is it important to be able to analyze these proteins? Like, it's really cool that we can visualize them with GFP and antibodies, but like, visualizing things doesn't improve meiosis. So go back to this slide. The main focus of the lab right now is to try and increase the crossover rate. And the tools that I worked on will help us analyze if we actually did that successfully. So one of the other projects that I was sort of involved in is trying to mimic the crossover phenotype of this mutant called ZMet. So as you can see from this chart, ZMet has a shifted crossover phenotype from wild type, as well as actually a higher crossover rate. And the main difference between ZMet and wild type is it's a hypomethylation mutant, so it has less methylation than wild type. So we're trying to mimic that using artificial demethylation. So we're going to deliver a methylation inhibitor, such as 5-azactidine, to Mays before meiotic S phase. So that's when the DNA divides before it undergoes meiosis. Right now, we're just testing a bunch of different delivery methods. Right. So was I successful in not being bored? I think so. As you can see, I learned how to do a bunch of different things. And I also had the opportunity to interact with a bunch of intelligent people in my lab, as well as at the seminars with the other interns. And I also had the opportunity to be a little bit more self-directed than last summer. Right. And none of this would have been possible without the help of my mentors, Matthews and Misha, as well as Professor Pulowski, as well as BTI and Cornell for hosting me, and NSF and USDA for giving me funding. All right. Thank you. Any questions?
Considering how conserved this function is and all the blast work that you've done, did you come across other relatives of the high-pen that may be of interest to target with this? Because I'm sure there's more than one.
In Mays?
Yeah.
So other than those two homologs that I showed you, there wasn't anything else. Because actually, apparently what happens, the reason why MLH3 doesn't have these homologs is because Mays is really good at getting rid of these extra meiotic recombination proteins for some reason, because they must interfere with how meiosis happens. So there weren't any others than these, and these, I mean, we could target these, but I don't know.
Okay, so if there's no further questions, we actually have now a short break to interrupt our 3-hour morning session. So if you want to go out, please come back. We're going to start 10:40.